This video follows up on our discussion about high-capacity vectors. In this one, I will focus on yeast-derived vectors and artificial chromosomes. There are a few considerations when using yeast as a host for cloning vectors. I will limit our scope to Saccharomyces cerevisiae, also known as Baker's yeast. There are three considerations. The first is the structural difference in the organization when compared to the bacterial host. In bacteria, there is no boundary to encapsulate the genomic material. So if you have plasmid in the bacteria, it enjoys a free exchange of enzymes required for DNA replication and transcription. In contrast, yeast has a nuclear membrane that compartmentalizes all transcription and replication-specific enzymes. So the vectors or plasmid cannot stay in the cytosol because then it cannot be maintained. So all vectors in eukaryotes, or yeast in this case, need to be present inside the nucleus, which limits the space available. In bacteria, the vector enjoys all the real estate a cytosol can provide. The limited space in yeast also means that vectors are packed in a crowded nucleus with the host genome. Second consideration, all yeast plasmid, well, most if not all, are E. coli-based vectors. This means you start out by expanding the vector in bacteria and then move them to the yeast. You do this because handling bacteria is much easier, the extraction of the plasmid is easy, and the DNA yield is typically very high. Now what we have just said is that there are two different host species for the same plasmid vector. This type of multi-host vector is called shuttle vector. This is the example of bacteria yeast. You can also have bacteria mammal, bacteria something else, or any two or more species. The point being that you need at least two different species to qualify as a shuttle vector. Third consideration is around selectable markers. We have seen the use of antibiotics as markers in bacteria. Yeast actually develop resistance towards antibiotics over time. This is because they spontaneously mutate towards resistance. Yeast also tends to be naturally resistant to many antibiotics. So instead we rely on auxotrophy, which is defined as the inability to synthesize an organic compound required for growth of the organism. Therefore, the selectable marker in yeast is linked to metabolism. The idea is pretty simple. The host yeast typically carries a mutation in a gene that is required for metabolism of a specific compound. But you can overcome the mutation if you put a vector containing the auxotrophy marker. So the vector complements the mutation in the host. This idea is similar to the LAC-Z alpha used in blue-white screening. As an example, the host is mutated in the URA3 gene, which is required for the uracil synthesis. If you use this mutated host, the vector then must contain the non-mutated version of the URA3 gene, so it can complement the host. So we say that the host is auxotrophic for the gene product required to make a specific organic compound. This product is typically an enzyme involved in the metabolism. We just saw the URA3 gene, which makes an enzyme required for uracil synthesis, but there are many more. Remember that the host is always mutant for the marker gene. Here's a short list of auxotrophic markers that people typically use. Most of these markers are genes responsible for amino acid synthesis. Hopefully you understand the basics. Now we can go back to vectors. I will focus on 5 vectors. Here's a summary of classic yeast vectors before I jump into details. A category on its own is a non-shuttle vector called yeast integrating plasmids. This is simply a bacterial plasmid with a yeast marker. The other class is a yeast episomal plasmid, which is derived from the 2-micron plasmid. The episomal plasmids have the 2-micron origin of replication and the bacterial origin of replication, making it a shuttle class vector. If you remove the 2-micron origin from the episomal plasmids and add the yeast ARS sequence, you call them yeast replicating plasmids. The presence of ARS and the bacterial origin means that it is a shuttle vector. To the replicating plasmids, you can add a centromere, and these are now yeast centromere plasmids. If you add telomeres to the centromere plasmids and linearize them, you obtain a linear yeast artificial chromosome. While centromere plasmids are shuttle vectors, linear YACs, because they're linear, are not shuttle vectors. From the linear YAC, you can take away the telomeres and add back components. This results in a back YAC hybrid or a circular YAC, which is a shuttle class vector. Let's dive into their details, starting with the yeast integrative or integrating plasmids. These are just bacterial plasmids with some yeast DNA. They were discovered or invented when scientists were trying to figure out yeast transformations. The DNA in question was typically a gene, which is also an auxotrophic marker. Same principles again, that you need a host with a mutated gene. The LU2 is involved in making the leucine amino acid, so unless you feed the yeast leucine, it cannot make leucine by itself. The original experiments were simple in that they took a PBr322 plasmid and inserted the marker genes in this plasmid. Given a leucine example, this gene is LU2. Note that the PBr322 has a bacterial origin of replication. You can insert this plasmid into the yeast and the leucine 2 of the plasmid will complement the yeast mutation, meaning that the yeast gains the ability to make the amino acid leucine. This bacterial plasmid with LU2 gene is called yeast integrating plasmid. Because of the bacterial origin of replication, it can only be propagated in a bacteria because the ori of prokaryotes is not compatible with the enzymes and DNA replication initiator proteins found in yeast. Since it cannot replicate in yeast, the plasmid will be lost after a few rounds of cell cycle, meaning that the complementation is lost, and because the leucine cannot be made, the yeast will also die. However, scientists noticed something interesting. A small population of yeast unexpectedly survived, suggesting that the complementation was still somehow active. This is not because the plasmid is lost. Interestingly, at a very low frequency, the plasmid integrated into the yeast genome, hence the name yeast integrating plasmid. This occurs through homologous recombination, or in this case an illegitimate crossover event, which works identically to the meiotic recombination. So what is the magic here? Well, the yeast genome has the LU2 gene with some mutations at a specific parts. The vector you provide has the same LU2 gene but without any mutation. So both of the genes are identical except for the mutated region. This identity is enough for a recombination event. As a result, the entire plasmid is integrated at the homologous region in the yeast genome, so the yeast becomes permanently complemented. It has fixed its auxotrophy. This means the only way to propagate any DNA contained in the integrative plasmid is through integration in the yeast genome. Since the original vector can only propagate as a vector in bacteria and not in yeast because it has to integrate, we call it a non-shuttle vector. Here are a few issues with the integrative plasmids. Since there are only 1 or 2 alleles of each gene, you can only have 1 or 2 copies of the plasmid integrated into the yeast genome. Now that it is integrated in the genome and not a separate thing, how the hell do you expect to isolate this DNA, especially if the copy number is low as well? On top of this, how do you screen for your DNA? Say you want to perform a restriction digest. If you cannot isolate the DNA, the digest will be performed on the entire genome and not just your specific DNA of interest. You see where this is going, right? Yeast integrating plasmids are terrible as vectors. They're great for long-term propagation of the DNA because you have chromosome level stability, but the practical utility of these plasmids is very low. Yeast episomal plasmids are a bit better. They are non-integrating plasmids based on the 2-micron plasmid. The 2-mu or 2-micron plasmid is a naturally occurring plasmid found in all baker's yeast. The entire plasmid has a bunch of interesting features, but the one we care about is the origin of replication. This origin allows the plasmid to replicate and propagate within yeast at around 25 to 100 copies per cell. A typical episomal plasmid uses the PBr322 plasmid, so it has the bacterial origin of replication. It has the 2-micron plasmid origin of replication as well, and an auxotrophic marker, for instance, URA3. Just to be explicit, there is one origin for bacterial propagation and another for yeast propagation in the same plasmid. The URA3 marker is for selection in yeast. So this vector can propagate in 2 hosts. It is a shuttle vector. Here's an example of an episomal vector. It uses all the components of PBr322. It has the antibiotic marker for propagation in bacteria, the PBr322 origin of replication for bacteria. In addition, it has 2-mu origin of replication and the URA3 gene. You can now use this plasmid to insert your DNA of choice, grow it in a bacteria, and then transfer it to yeast. The host will be URA3 negative because the vector has the complementing marker. Once the vector is inside the yeast, it can propagate at around 25 to 100 copies per cell because of the 2-micron origin of replication. The issue is that 2-micron plasmids also occupy the same space as the episomal vectors. If you think about it, you have the origin of replication as the homology between two plasmids. If there is a crossover, you can get recombinant plasmids, which are chimeras. The other problem is that episomal plasmids also have the probability that they integrate into the host genome. This is because of the URA3 gene, which is conceptually similar to the leucine situation we saw in the integrating plasmids. So episomal plasmids can become integrative plasmids. This is the definition of an episome, which is a plasmid or DNA that has a non-integrative form for the most part, but it can also have an integrative form. The next one is yeast replicating or replicative vectors. They have the bacterial origin and marker, but instead of 2-micron origin, they carry the yeast autonomously replicating sequence, which is the starting point of DNA replication in the yeast genome. Here again, you have two replication origins for two hosts, meaning that the replicative plasmids are also shuttle vectors. Yeast-derived origin has specific regions that are recognized by initiation factors to start DNA replication. I have a video linked in the description discussing these regions in much more detail. Intuitively, here's a representation of the vector. It is again based on the PBr322 plasmid. It has an antibiotic marker for selection in bacteria and URA3 for selection in yeast. It has yeast ARS and it can carry your specific DNA of interest as well. The usage is the same. You grow them in E. coli first, then move them to the appropriate host where a vector can complement the host. The host receiving the vector will survive even if you don't provide the yeast with an external uracil. All the yeast that do not receive the vector will die because the host cannot be complemented. A small side note, replicating plasmids were discovered as a part of a screening experiment in 1979 where scientists found that instead of integrating into the genome, some plasmid could replicate independently for many cell cycle generations. This was because of the ARS sequence. They didn't know that in 1979. The actual description and name of this DNA, the ARS, was given much later. Replicating plasmids are typically found in low copy numbers. They can also over time become integrative because of the homology through the URA3 gene or any other piece of DNA in the vector. The replicating plasmids are nice if you are trying to insert DNA that eventually makes proteins. The low copy number means not a lot of proteins can be made. Compare this to episomal plasmid where the copy number is somewhat high, means a lot more proteins can be made. And sometimes having a lot more proteins can be toxic to the cell. So replicating plasmids can be useful in such cases. But here are some problems with them. So you have the replicating plasmid in the host. Yeast divides through budding, but unfortunately, through some unknown mechanism, the bud fails to receive high amounts of replicating plasmids. So YRPs segregate asymmetrically, and the plasmid predominantly remains associated with the mother yeast cell. So after a few rounds of buddings, you can expect that the yeast transformants, or the mother cells, will dilute over time and the plasmid will be lost from the population. To be more explicit, in generation 1, the empty daughter cells buds more cells. The mother will bud again, but the plasmid will remain with the mother. In the next round, these mothers will bud again, but they're all empty. The grandmother here is the only one to have the plasmid, and no new cells contain a high number of replicating plasmid. So we have increased the yeast population size without increasing the number of replicating plasmids. Next up is the yeast centromere plasmid. It has everything that a replicating plasmid has, but in addition, it also has a centromere DNA sequence from one of the yeast chromosomes. If you recall the structure of chromosome in metaphase, the spindle assemble at the kinetochores, which form the centromere, which is formed because of the centromere DNA. So centromere plasmids are just replicative plasmids with the centromere-associated DNA. The 4 in CEN4 means that the region is derived from yeast chromosome 4. CEN4 region also was discovered in the same screen where some plasmid had long-term stability and proper segregation after mitosis. The strategy to use this vector does not change. Same principles follow here too. Because of centromere DNA, the plasmids have a specific chromatin structure and they quite literally behave like mini chromosomes. This gives them mitotic stability but drastically reduces their effective copy number. Unlike replicative plasmids, the daughter cells here receive centromere plasmids in equal proportion from the mother cell because now we have the centromere DNA in the plasmid. This segregation extends to meiosis as well, where a centromere plasmid follows Mendelian expectations. Just a crucial note that despite being called a mini chromosome, these things are still circular vectors. Okay, I've been using PBR322 origin for all these vectors, but that's because yeast vectors were originally developed before 1980s. PUCs and Bluescripts were invented in 1980s. So today, most vectors are optimized to maximize the copy number through PUC19 and Bluescript modifications. Now we have arrived at yeast artificial chromosomes. They are non-integrative that behave like linear chromosomes, quite literally. The YACs carry everything a centromere plasmid does. However, the bacterial origin is only meant to propagate the original plasmid in the bacteria. The final form of the vector cannot be propagated in the bacteria. This is because these vectors are used in a linear form. In bacteria, they will get destroyed by exonucleases. This important distinction that final form of the vector can only be used in yeast makes linear YACs a non-shuttle vector, even though it has the prokaryotic origin of replication. In all previous cases, the final vector can also be replicated in bacteria. Therefore, they're all shuttle vectors with a relatively small insert size. Linear YACs are meant for multi-100 kb or megabase size insert, thereby mimicking a chromosome. As I said, they have everything from a centromere plasmid, but in addition, YACs have telomeres. A little side note that these telomeres are built using DNA from a protozoan. Here is an intuitive idea of the vector. The starting YAC vector bears similarities to the centromere plasmid. It has the PBR322 components, the ARS and SEN4. In terms of auxotrophic marker, it has 4 different ones. The SUP4 usually carries 1 or 2 cloning sites. The His3 is surrounded by BAMH1 sites. And flanking the His3 are 2 telomeric sequences. This is the starting YAC vector. To use this, you bring a genomic fragment digested by EcoR1. The vector is opened by BAMH1 and EcoR1. This splits the vector into 3 parts. The His3 portion is discarded and the other 2 portions are kept. In this, you can notice that both sides have an auxotrophic marker. The SUP4 is split and the telomeres are at the ends. In between these two, your DNA can be inserted. This final vector is not a circular fragment. The final vector has 2 markers, which means that the host must have 2 mutated genes. That way, the final yeast will have 1 large fragment complementing 2 mutations. Let's talk about this split SUP4. SUP4 is a suppressor of the mutation in one of the genes involved in purine synthesis called ADE2. The short end of the story is that if you have split SUP4, as you do by inserting a fragment in the vector, the host ADE2 mutation cannot be suppressed, and the cells that have insertionally inactivated SUP4 accumulate a red pigment. This is a way to screen for yeast that have a YAC. Typically, the longer the genomic fragment, the more stable it is in the YAC form. A side note, you can also have TRP1 on both sides or URA3 on the both sides. Since the host is negative for at least 2 marker genes, these type of YACs containing yeasts will die, and only the correct ones live around for about 1 or 2 copies per cell. Unfortunately, YACs are terrible vectors. Even the Human Genome Project eventually stopped using them for making human genome libraries. These issues I will talk about are largely associated with genome mapping. The first issue is chimera clones. The expectation is that you have a single insert in the vector, but in practice, about 10 to 60% of the clones have 2 inserts. If you're doing genome mapping, having 2 inserts in a single library will give you misleading information. Internal deletions are also very common. Sometimes complex regions of about 20 to 300 kb sizes can be deleted from an insert. This can occur due to mitotic growth, or recombination events with the host genome, or even internal to the fragment itself. YACs are also unstable if you grow them for too long, and often you can lose them. The biggest issue is the isolation of a YAC. Yeast genome is about 12 megabases. It has 16 chromosomes that range from 250 kb to around 1.5 megabase. The size range of a YAC is exactly the same as the size of a typical yeast chromosome. then how do you isolate a YAC and not a yeast chromosome? These are a few reasons why YACs were abandoned. The Human Genome Project eventually switched to BACs and PACs. BACs primarily, which are also big, but at least the bacterial genome is in single copy and quite a bit large, so the isolation of BAC DNA isn't as challenging. A slightly better YAC is a circular YAC. This uses bacterial artificial chromosome components. Here's a rough bellow-back structure that you may recall from my video on BACs. Scientists added a few things to this bellow-back vector. For simplicity, I will call this entire piece of DNA the bellow-back. To this back, we have added history marker, neomycin selection gene, and a centromere DNA from yeast chromosome 6, and a couple of regions that function as homology regions. These homology regions are homologous to the part of a YAC vector, and the homologies flank the ARS in this yeast vector. By recombination of these two pieces of DNA, a larger circular fragment can be obtained. This looks exactly like the starting vector, but now we have an ARS in between the homology regions. This is a BAC-YAC hybrid vector, also called a circular YAC. Through the recombinations, we have avoided the incorporation of telomeres in the final vector. This remains as a circular DNA. It works as a shuttle vector because it has the F plasmid origin from the bellow-back and the ARS from the linear YAC. The isolation of circular YAC is slightly easier because you can do alkaline lysis and avoid getting the genomic DNA. Circular DNA is also much more stable and resistant to shear damage. Even though we have this BAC-YAC hybrid, people still prefer BACs for most application. They're just generally better unless there's a specific reason to use a YAC. And this sums up our discussion on yeast cloning vectors. I hope this discussion was useful, and as always, I will see you in the next video.